Chapter 7, Microbial Genetics. In this chapter we're going to cover the structure and replication of genomes, gene function, mutations of genes, and genetic recombination and transfer. So for the first topic we're going to talk about the structure and replication of genomes. The learning objectives for this topic are: compare and contrast the genomes of prokaryotes and eukaryotes, describe the structure of DNA, and discuss how it facilitates the ability of DNA to act as genetic material. Describe the structure and function of plasmids. Compare and contrast prokaryotic and eukaryotic chromosomes. Describe the replication of DNA as a semi-conserved process. Compare and contrast the synthesis of leading and lagging strands of DNA replication. The subtopics in this topic are the structure of nucleic acids, structure of prokaryotic genomes, structure of eukaryotic genomes, and DNA replication. So here's a reminder of what the structure of nucleic acids are. Now for DNA we have 4 nitrogenous bases. We've got guanine and cytosine, adenine and thymine. In RNA we also have 4 bases, but thymine is replaced by uracil. So in RNA we would have guanine, cytosine, adenine, and uracil. As far as bonding goes, guanine always goes with cytosine, adenine goes with thymine in DNA, adenine goes with uracil in RNA. Now what do we mean by goes with? So remember that a nucleotide is composed of a sugar, a phosphate group, and a nitrogenous base. The sugars and the phosphates of the different nucleotides are hooked together from the 5' carbon that holds the phosphate group to the 3' carbon, which normally holds a hydroxyl group. Once we have performed a dehydration reaction to hook these together, we lose that hydroxyl group. So we have phosphate sugar, and the nitrogenous bases stick out. Well, in DNA, which naturally forms a double strand, we have another strand running anti-parallel. So this one is going the other direction. You notice the sugars are upside down. And the bases are sticking out, and they hydrogen bond with the nitrogenous bases on the other strand, and this keeps the 2 strands together. Now, we call this anti-parallel. This one's running from 5'. On the end, we've got our phosphate on our 5' carbon, going down to the 3' end. This one's going in the other direction. This arrangement is very good for allowing DNA to replicate itself. Hydrogen bonds are a whole lot easier to break than covalent bonds, so the strands can be separated and new matching nucleotides can be brought in, and we would end up with exactly the same molecule of DNA, which we'll be going over later on in this topic. On to the structure of prokaryotic genomes. As you remember, prokaryotes do not have a nucleus. They do have a nucleoid, which is an area in which the chromosome is concentrated. So if we take a look at the nucleoid and we take a look at the chromosome, this is what we're going to see in a prokaryote. We would see a central protein scaffolding, and we would see loops of chromatin that are called a loop domain. Well, if we look at a blow-up of this loop, we would see that their DNA all winded up. And this is what it looks like. We have a protein here that gets wound with the DNA. Now, this protein, don't confuse it with histones. Histones are in eukaryotes. We're going to talk about that next. So, if we take a poor little E. coli cell and we pop it so that its chromosome comes out, you can see these big old loops that have been unwound from the scaffolding. So you can see that they come in loops. I also like this micrograph because it shows a plasmid. A plasmid is a small loop of DNA that is extra chromosomal. It is outside the chromosome of the bacterium. And we'll be talking more about plasmids when we talk about horizontal gene transfer. For the eukaryotic genome, we're going to go from smallest to biggest, so the opposite direction that we went for the prokaryotic genome. Eukaryotic DNA is wrapped around histones. Histones are unique to eukaryotes, although archaea have histone-like proteins. We have different subunits of histones that come together to make this cube, and we wrap the DNA around it. Now we have another histone subunit that stabilizes the linker DNA. Now we take these, we call these nucleosomes, and we wrap them up still further into chromatin fiber. Now, if genes are actively being transcribed, then the area of DNA where those genes are remain as chromatin fibers in loosely packed euchromatin. Now, if we're not using the genes, that area gets more tightly packed. And when the cell is getting ready to divide, it gets highly condensed into our familiar chromosomes, which reside in the nucleus unless the cell is undergoing mitosis or meiosis. Speaking of cell division, before a cell can divide, whether it's a prokaryote or a eukaryote, you have to make a copy of the DNA. You have to make a copy of the genome because the new cell needs a copy as well as the old cell. So we're going to go over DNA replication in prokaryotes. That's where it was first intensively studied, and then I'm going to mention a few differences that show up in eukaryotes. So first off, when DNA is being replicated, when we are making a deoxyribose nucleotide polymer, then we always start out with the triphosphate version of that nucleotide. So for guanine, we would have guanine triphosphodeoxyribonucleic acid. Okay? The reason being that when we hook this in, these two phosphate groups are snipped off, and that provides the energy for the polymerization. So it's kind of like the nucleotides are bringing their own ATP. In fact, for the adenine, it is ATP. The DNA polymerase, and we always go from 5' with the phosphate to 3'. That's just the way the active site of the DNA polymerases are shaped. So we have an existing DNA strand that we're lengthening. We bring in a triphosphate nucleotide, comes in, we snip off the diphosphate, and we use the energy for synthesis. And we just keep going along like that.